Today we are going to finish up chapter 18, and then we will move on and start chapter 21. It'll give you a little bit of an introduction to where we're going to be in lab next week. For chapter 18, there are 3 topics that we need to, that we'll cover in order to finish off chapter 18. We'll go through control of blood flow, velocity of blood flow, and then we'll look a little bit at something called bulk flow. So those are those 3 topics. And then, like I said, we'll just introduce the structures of the upper respiratory tract, and that's actually pretty quick, and you're familiar with a lot of those already. Okay. So blood flow, to me, it's like Goldilocks, just right. It's not too much, it's not too little. It's finding that balance that is just right. And your body does a really good job at the local level of perfusion at just the right level. There's kind of 3 levels. There's like a hierarchy of control that we'll talk about. And each one can kind of override the next to the next to the next. So because we have these multiple levels of control, it allows for very precise perfusion per tissue. So the purposes of tissue perfusion are to deliver oxygen and nutrients to the cells, as well as to remove waste from the cells. At the level of the lungs, we'll exchange gases. At the level of the digestive tract, absorption of nutrients. And then for kidneys, we've talked about kidneys a little bit. Blood flow is going to be the major factor one of the major factors in urine formation. So blood flow is controlled by extrinsic factors as well as intrinsic factors. We've actually already looked at the extrinsic factors because those were the ones that affected total peripheral resistance and maintaining blood pressure. So I'm going to skip forward a little bit to this little graph, and the extrinsic factors are here in the peach colored boxes. We have vasodilators, vasoconstrictors. For extrinsic controls, those were those short-term neural and short-term hormonal controls. So for the neural controls, remember, it's always sympathetic nervous system. And then these are vasomotor fibers of the sympathetic nervous system that control vasoconstriction or vasodilation. And of course, we have those reflexes, baroreceptors and chemoreceptors that are going to influence the rate of vasomotor firing. So extrinsic, we have our sympathetic vasomotor fibers, as well as the receptors that are influence them. And then we have those hormones that we talked about. And we have for vasoconstrictors, angiotensin 2, ADH, because it will reabsorb water in those kidney tubules. Remember, there are 4 functions of angiotensin 2, but we'll skip those for now. Epinephrine and norepinephrine are also vasoconstrictors. And then we had our lone holdout for atrial natriuretic peptide as a vasodilator. So this is actually a very good graphic to study because it summarizes the extrinsic controls and then the intrinsic controls as well, which we haven't gone over, but that's where we're going. Okay. So we covered extrinsic when we covered total peripheral resistance on Monday. And then today we're going to look at intrinsic, which is also called autoregulation. So this is on a actually less than second by second basis as far as what the tissues that are fed by a capillary bed, what's happening at that level. So there's a lot of local control because they have to have the last word. As far as, sympathetic can say, hey, I really want to shunt blood from the digestive system to the skeletal muscle system. But eventually the digestive system is going to say, hey, I need a little bit of love here too, and need to take over with this autoregulation. Okay. So local arterioles that feed capillaries and then feed tissues supplied by that capillaries can modify their own diameters. They do this in two ways. There are metabolic controls and myogenic controls. Metabolic controls are what's going on at the level of the tissue. If there is a decline in the level of oxygen or an increase in the level of carbon dioxide or hydrogen ions. So hydrogen ions can be released with lactic acid and some other metabolic reactions will cause our acidic products. Carbon dioxide is also acidic as well. And then we have potassium ions are going to indicate that we're starting to get an ion imbalance. And then adenosine is a very potent hormone, as well as prostaglandins. We'll talk about nitric oxide as well. And some of these are more regulators more in one tissue than others. So the brain is very sensitive to carbon dioxide. The heart is going to use adenosine when it needs to. The hierarchy of these is a little dependent, is tissue dependent. Okay. But when we get any of these occurring, then we'll get vasodilation. We'll get vasodilation to increase blood flow so we can get more oxygen, we can get rid of the CO2, or adenosine is used by the heart when it needs additional oxygen as well. These can have a direct effect on those precapillary sphincters, or they can cause the release of nitric oxide. And nitric oxide is a very powerful vasodilator, but it goes away very quickly too. But it has the ability to override the sympathetic nervous system, albeit momentarily. So very powerful, but dissipates very quickly. So metabolic, okay, so we're gonna go back. So for intrinsic, there are two types: metabolic and myogenic. Metabolic is going to deal with the metabolites produced by the tissue metabolites. So a decline in oxygen or increase in carbon dioxide, hydrogen ions, decline in pH, potassium, adenosine, prostaglandins. Both of these will trigger local vasodilation, either directly or through the release of nitric oxide. Endothelins are potent vasoconstrictors. They can be released by the endothelium. Sometimes this is due to damage or sometimes other signals, but nitric oxide can overcome endothelin, because nitric oxide is saying, hey, I need blood flow now, or like the cell's gonna die or it's gonna be damaged. So nitric oxide is going to kind of have that one of the last words. For metabolic controls, I am more concerned with this slide. Okay. What they are, so declining O2 and increasing these, and then their effects. So this slide is important. This one, more informational, just that it's balanced. We have vasoconstrictors as well. Then we get to myogenic controls. So myogenic, if you remember from 1141, myo is one of the words that means muscle. When we're talking about a myogenic response, we're talking about the vascular smooth muscle in the arteriole. When there's an increase in blood pressure, the vessel wall stretches, and then that smooth muscle is going to respond by constricting. hierarchy goes that we have the sympathetic nervous system. Normally, we're in that middle rate of between vasoconstriction and vasodilation. We get chemoreceptors, baroreceptors, or we exercise, or there's some alteration to that, and we go, okay, let's vasoconstrict more, or let's vasodilate more. That's kind of this top hierarchy. Then we get to the actual tissues, and they're going to need second by second control, and that's determined by those metabolic factors, with nitric oxide saying, I trump pretty much everything else. With an increase in blood pressure, so sometimes so let's say we have vasoconstriction and there's increase in blood pressure, arterioles need to have a protective mechanism in place. Like they can only take so much pressure before they were to burst, and that would be bad for everything. Okay, the stretch myogenic response is going to protect smooth muscle from getting over distended, because when you stretch it too far, then it's going to cause it to vasoconstrict, to take off that pressure against the wall. Reduced stretch decreases mean arterial pressure, and it's going to respond by dilating. Don't worry.
I have a question.
Yeah.
So in like the simplest of terms, like a metabolic response is going to vasodilate, and a myogenic response is going to vasoconstrict?
Usually, yes. That's
Okay.
the directions that they're going to go.
Great, thank you.
As far as change. And then they'll go back. So like if we're looking at metabolic, let's say we're exercising and we're producing a lot of carbon dioxide and hydrogen ions, that stimulus is vasodilation. So usually we go, okay, let's vasodilate and perfuse this tissue. But then it will, after that need is done, it will go back to that middle level.
Got it. Okay, thank you.
It won't stay vasodilated. And then yes, on the myogenic side, this is kind of more on the protective end, where if we're getting too much pressure stretch, it's got to stop. So it's going to vasoconstrict. And then when the pressure decreases, it will go back to its normal size. Okay. So again, we're back to this nice little diagram. I like it when it puts everything kind of together and it's not too busy. So for intrinsic, we have metabolic, which this is actually leading right into your question and showing that metabolic stimuli tend to vasodilate, and myogenic tend to cause vasoconstriction. So this is exactly answering what you're asking. So blood flow, we've talked about local, and then we're going to talk briefly about how it changes in special areas. For skeletal muscle at rest, myogenic and neural mechanisms dominate. So the sympathetic nervous system, with exercise, then blood flow is going to need to increase to that area. And then local controls are going to override sympathetic. Again, it's that hierarchy, but then with activity, we get all of these metabolic factors building up, and that's going to cause local metabolic intrinsic regulation to override sympathetic. The brain actually has a very consistent flow rate. It doesn't change very much. Part of this is because it has a high need for nutrients. It doesn't store nutrients. So it doesn't store glycogen or triglyceride like a lot of our other cells do. We pack it full of neurons, but it means that it needs to have a pretty consistent oxygen and nutrient delivery. Most potent metabolic stimulator in the brain is going to be carbon dioxide. If the carbon dioxide level gets too high, we actually go unconscious, but within normal ranges, carbon dioxide is the one that is going to cause vasodilation. There are some myogenic controls. This is because you don't want to have large changes in arterial pressure in the brain. They are susceptible to rupture. The capillaries are pretty fragile. When the mean arterial pressure falls below 60, it can cause fainting. And when it's above 160, it can result in cerebral edema. And we'll talk about why that is. This is going to cause an increase in what's called hydrostatic pressure. And the brain doesn't have like the lymph drainage that like the rest of the body has. So it can swell, and because it's encased in your skull, there's nowhere for it to go. So it's a very bad cascade. So the brain works very hard to keep constant flow and constant pressure. And flow is going to be primarily dictated by this increase in carbon dioxide, and maintaining that pressure will be under sensitive myogenic control. So these two work together. The skin supplies nutrients to the cells, it provides a blood reservoir, but the big one is that the skin is going to help regulate body temperature. There are extensive venous plexuses below the skin surface. And this is going to be controlled by their thermoreceptors in your hypothalamus. And then as it monitors body temperature, then the flow through these plexuses are controlled by the sympathetic nervous system. So the skin is very important in maintaining body temperature. That flow rate in a plexus can range from 50 mils to 2,500 mils per minute. And it's interesting, like, so when it's really cold and the flow gets cut off, the reason why people have rosy cheeks is because blood gets trapped in the little loops. So that's also in your textbook. It's kind of, sometimes your textbook has fun examples. Okay, so you guys are probably wondering, like, what do I need to remember? So skeletal muscle at rest, we'll just say the sympathetic nervous system dominates, and then with activity, local controls override due to this high change in metabolic activity. So start with sympathetic, and then after with exercise, we switch over to local metabolic. The brain uses both metabolic and myogenic to maintain a constant flow rate and a constant pressure. And that main metabolic control is going to be carbon dioxide. For the skin, the one I like to focus on for right now, because you've already done integumentary, is the role in helping to regulate body temperature, and it's controlled by thermoreceptors in the hypothalamus, and then that relays through the sympathetic nervous system. Okay. The lungs. The lungs is, we're going there next. We're going to the respiratory system. So I'm not going to say too much about the lungs right now, but the pulmonary circuit is short and it's low pressure. Okay, so 24/10 versus 120/80. And so the autoregulatory mechanisms are opposite. When oxygen levels are low, normally if that happened at the skeletal muscle, we'd have vasodilation to flood that area with oxygenated blood. But just like everything in the lungs, which is backwards, you've got, deoxygenated pulmonary arteries and oxygenated pulmonary veins. Autoregulation is opposite as well. Low oxygen levels cause vasoconstriction because they want to force. Normally, we only run blood for air exchange through the bottom section of our lungs. When we vasoconstrict, we push blood up into all of that lung volume, and it oxygenates it better. So in the lungs, low oxygen levels cause vasoconstriction so that we can better perfuse the lung tissue and get oxygen levels up. Okay, so everything's backwards in the lungs or opposite. For the heart, the thing about the heart is the heart has something called myoglobin. It's kind of like hemoglobin, but it is you're going to be specialized to maintain, and skeletal muscle fibers, red ones have this too, but myoglobin stores oxygen inside the cardiac cell, and normally this is sufficient. During exercise, the oxygen demand is increased, and so the coronary vessels are going to produce adenosine, which is a very potent vasodilator. So for the heart, myoglobin is usually sufficient, but to sustain it through ventricular systole, but with exercise, it will produce adenosine, so a local metabolic control, and that will vasodilate those vessels. Okay. Now we're going to look at velocity of blood flow. Velocity of flow changes as blood travels through the systemic circulation. It is fastest in the aorta and slowest in the capillary, and then increases in the veins. Speed is inversely related to total cross-sectional area. So right now we're looking at arteries versus arterioles versus capillaries. So we're looking at segments of vessels. Capillaries have the largest area, so the slowest flow. This is going to allow time for exchange between blood and tissues. Vasomotion is the intermittent flow of blood through capillaries. So the precapillary sphincters are kind of always going on and off and on and off. So the flow kind of has a little bit of a rhythm to it in the capillaries. It's interesting, not maybe very exciting. Okay, so this, remember, speed is inversely related to the total area of the vascular bed. So the aorta is like 2.5 centimeters squared. And then when we get up to all of the arteries, I think we get up to about 500. Okay? So the total area is very low. And so the blood has to flow very fast through it. Because remember, the amount of blood is staying the same. So if you don't have much area, you have to get that through very quickly. But it has a very large lumen, so there's not a lot of resistance, and there's a huge pressure gradient. At the level of the capillaries, so we branch from arteries to the aorta to arteries to arterioles, and then we get to these capillary beds, the area greatly increases, up to about 4,500 squared centimeters. So a lot of area. And so now that blood has branched out, like we were traveling on this gigantic freeway, and now we're off on these itty bitty teeny tiny country roads. And we're having to go really slow and really carefully through them. So the speed drops off. And then as the surface area decreases, the speed picks back up again, but we don't have the same pressure gradient driving it. But right now, let's just focus on area and blood flow. So capillaries have the highest area of vascular bed and the lowest blood flow, so the lowest rate. So the most area and the slowest flow to allow waste to be exchanged and nutrients to be exchanged. How do molecules across the capillary. So right now we're going to look at molecules because we're moving on to fluid. So your book says 4 different routes. To me, 2 and 3 are about the same, but the 4 different routes are diffusion, simple diffusion directly through the endothelial membranes. And if you go way back to like when I first met some of you, these are going to be small, non-polar, such as respiratory gases, oxygen and carbon dioxide, as well as lipid soluble molecules because the plasma membranes are lipid soluble. So simple diffusion requires no energy, but it does go down its concentration gradient. Simple diffusion is the fastest way to get through. So because they just go straight through on that gradient. The second and third way, which I usually put together, because it just depends on the type of capillary. So it's not really a different type, it's just different type of capillary, are to either pass through clefts or to pass through fenestrations. These are water-soluble solutes, such as amino acids and sugars. So these are polar molecules that cannot pass through diffusion. The fourth way is active transport using pinocytotic vesicles, and these are going to be for larger molecules, larger proteins. This just is a nice little graphic. Again, I like it. Simple diffusion through the plasma membrane for carbon dioxide and oxygen and lipid-soluble substances. 2 and 3, either moving through clefts or through fenestrations, bigger spaces, pores, for amino acids and glucose, or active transport of pinocytotic vesicles for larger proteins. Okay, so bulk flow. So 20 liters is forced out of clefts and capillaries at the arterial end each day. We only have like 5 to 6 liters of blood. So if we did not reclaim this, we wouldn't survive a day. So it's important, extremely important, to learn how fluid is getting out of capillaries and then how it's getting recaptured, because we need to recapture that fluid in order to maintain plasma volume and cardiac output. Okay. Bulk fluid flow is a continuous mixing between plasma and interstitial fluid, and it is determined by 2 opposing forces: hydrostatic pressure and colloid osmotic pressure. And there are 2 of each of these. There is a hydrostatic for the capillary. There is a hydrostatic for the interstitial fluid. There is an osmotic for the capillary and an osmotic for the interstitial fluid. So there are these pressures, but because we are looking at the movement between capillary and interstitial, each location has a hydrostatic and an osmotic. Hydrostatic pressure is the force exerted by the fluid pressing against the walls. So it is determined by that pressure gradient. Capillary hydrostatic pressure is going to be your greatest force. So capillary blood pressure forces fluid through the capillary walls. This is very important. It is greater at the arterial end than at the venual end. Okay, so there is a drop in pressure between the arterial and the venual. The hydrostatic pressure of interstitial fluid, so the pressure of the interstitial fluid trying to push fluid into the capillary, is usually assumed to be 0 because lymphatic vessels are draining the interstitial fluid. if it's assumed to be 0, why do we bring it up at all? Because it's assumed to be 0 when the lymphatic vessels are working. But there are conditions where lymphatic vessels are removed or not functioning, and then this is no longer 0. But we're going to look at a normal, healthy individual when we're calculating net filtration pressure. Colloid pressure is created by, colloid osmotic pressure is created by plasma proteins. So these plasma proteins cannot get out of the capillary, and so they are encouraging osmosis of water back into the capillary. It's going to stay consistent because plasma proteins don't leave the capillary. So the osmotic pressure of the capillary is around 26. Interstitial fluid osmotic pressure is considered inconsequential because interstitial fluid has very low protein content, and it's considered to be around 1. Net filtration pressure comprises all forces acting on a capillary bed. So we have forces that push or pull fluid out of the capillary. And those forces are the hydrostatic pressure of the capillary and the osmotic pressure of the interstitial fluid. In this hydrostatic capillary pressure in arteries is going to be around 35. In veins, it'll be about 17 in the venous system. Osmotic interstitial is usually around 1. Then we have forces that push or pull fluid into the capillary. And those are going to be the osmotic pressure of the capillary, which is consistent at, does anyone remember the number?
0?
No, it's the next one. This one's a big one. This is because plasma proteins stay in the capillaries. It's 26. Okay. And the way you can kind of, well, let me finish typing, then we'll talk. And then the hydrostatic of the interstitial. This is the one that's usually, as long as lymphatics are working, is usually around 0. Okay. The capillaries are going to be the controllers of net pressure, net filtration pressure. So they're going to have this hydrostatic pressure is higher on the arteries and lower on the veins. Osmotic stays the same. And if you notice, it's kind of in between this hydrostatic pressure of arteries and this hydrostatic pressure of veins. And that's going to become important So the combined value for the force going out of an artery, okay? So the force out of an artery is 35 plus 1. So we're going to look at out at the artery is 35 plus 1. The pressure in that's trying to force water in at the arterial side. So these are all happening at the level of the capillary bed. It's just, are we on the arterial side or are we on the venule side of this capillary bed? So we have, we have this capillary bed, and we have the arteriole leading in, and we have the venule leading out. So we're going to have fluid moving out on the arterial side and moving in on the venule side. The question is, why does this occur? And it's because you are going to look at the forces out minus the forces in. So on the arterial side, the pressure that's out is 36 millimeters mercury. The pressure that is trying to push fluid in from the interstitial into the capillary is 26 plus 0, so 26. For net filtration pressure,
I have a question.
Yeah.
So before you move on from the out and the in, so do we not take into consideration the 17 on the venous end?
We just haven't gotten to, so you'll need one set of calculations for the arterial side, and then one set of questions for the venule side.
Oh, okay, got it.
Calculations. So the same thing that we're doing right now for the arterial side, we'll do again for the venule side. So net filtration pressure is going to be, you're going to look at 36 minus 26. And that will equal 10. So we're going to do, this will be 10, a pressure of 10 out. So 10 millimeters of mercury difference pushing fluid out, which is how we end up with 20 liters of fluid in the interstitial per day. Then on the venule side, we're going to do the same thing. The pressures out are now going to be that 17 plus 1. Because the pressure has dropped as we've gone across the capillary bed. So now the pressure pushing fluid out is 18. The plasma proteins haven't gone anywhere. They're still in the capillary. So they have the same pressure. They're still pulling in at the same rate. So in is 26 plus, under normal conditions, 0 for 26 millimeters of mercury. Okay. So then on the venule side, we have a net filtration pressure of equals, okay. What minus what? It's not 36 anymore. We're down to
18.
Mhm. And then we still subtract 26. So now we have -8. And when you get a negative number, it means that there is a sucking in. I don't know why, but it's like an engineering term. They do push and not pull. They do push and suck. I don't know. But then, so this -8 means that fluid now gets pulled in. Okay, which one is bigger? Artery, the push or the pull?
Push.
Right. So push pushes out 20. Pull only pulls back about 17 liters, 17 to 18. Where does the other 3 liters go? They go to the lymphatic system. Okay. So it's not perfect. And that's actually good because we need flow through the lymphatic system as well. So for 2 categories of forces, hydrostatic, which is the pressure. What is that pressure gradient pushing on the walls to push fluid out or push fluid in, which is not very effective. And then, so you'll have a hydrostatic for the capillary, and then osmotic for the capillary, that's the plasma proteins. And then the tissue osmotic and hydrostatic tend to be very minimal. They don't really, unless you're under a disease process, they kind of fall out of the equation. But if you are given hydrostatic and osmotic pressures, you should be able to calculate a net filtration pressure and determine if it's going out of the capillary or into the capillary. If it is a positive number, it's going out of the capillary. If it is a negative number, it's going into the capillary. So if I told you that the capillary has a net filtration pressure of -7 millimeters of mercury, is it on the arterial side or is it on the venule side?
What was the number again?
-7.
Annual.
Correct. Okay. So we have positive numbers on the arterial side. We have negative numbers on the venule side. We have fluid moving out on the arterial side. We have fluid moving in on the venule side. And I'm going to save that so I can post it. Make it, it's not too messy. Okay. Yeah, this one may take a little bit to sink in, but if you draw it out, it's not terrible, right? You're like, sure, yes, it is. Okay. So we have calculating net filtration pressure. You take the pressures that are pushing out, which this is going to be the big one, hydrostatic pushing out, minus the pull. So this is push out minus the pull back in. capillary is going to be, what's going on in the capillary is really what you're concerned with. I only mention these because they can change with disease processes. But for a healthy individual, it's the hydrostatic pressure at the capillary minus the osmotic pressure at the capillary. Which one of these changes? Hydrostatic pressure at the capillary or osmotic pressure at the capillary? Between the arterial and the venous end.
Wouldn't it be the hydrostatic?
Yes. Okay. So that is very similar probably to the question that you will see. The hydrostatic pressure is what changes. At the arterial end, it's around 35 because you have more pressure coming from that arterial end. Then the pressure dampens down as we go along the capillary. And by the time we get to the venous end, it's down to about 17. Meanwhile, we have plasma proteins in the capillary. They are too big to go anywhere. So this number stays the same at around 25, 26. So the osmotic pressure stays the same because plasma proteins cannot escape the capillaries. I mean, there's a small amount through pinocytosis, but we're talking itty bitty. Okay. But in general, the big osmotic proteins in the plasma stay there. On the arterial end, the pressure, hydrostatic pressure, is higher than this osmotic pressure. So fluid leaves at the arterial end. At the venous end, osmotic pressure is greater than hydrostatic pressure. So fluid is pulled back into the venous end. Interstitial fluid is returned to the blood via the lymphatic system. So do you know which one changes, which one stays the same? And is fluid leaving? Is fluid being returned? So this is one of those things that you're going to need to diagram out and like draw in the little plasma proteins and draw in the arterial pressure against those walls, and it will become more clear. This is really hard to see, but it goes through the calculation that I did on the whiteboard. The take home from this one is hydrostatic pressure on the arterial end is 35. On the venous end, it is 17. So hydrostatic pressure changes. Osmotic pressure is going to stay the same because these plasma proteins are here and here and here. They're everywhere, they stay in the vessel. So it is going to stay the same. If you subtract osmotic from hydrostatic, you'll get a positive number on the arterial end. You'll get a negative number on the venual end. And here and here on the arterial side, fluid moves out. And on the venual side, fluid moves in. That actually ends the circulatory system. The part 3 is covered in lab. So the respiratory system involves 4 processes. This is a good diagram of those 4 processes. The first is pulmonary ventilation. And ventilation is going to consist of 2 phases: inspiration, moving air into the lungs, and expiration, moving air out of the lungs. And we'll go into both of those in much greater detail next week. Then we have external respiration, which is the exchange of gases at the level of the lungs. Oxygen goes into the blood, carbon dioxide goes from the blood to the lungs. Third process, transport of respiratory gases. Oxygen is transported to, CO2 is from the tissues to the lungs. And then internal respiration is the diffusion of oxygen into the cells and of carbon dioxide out of the cells. So very basic overview. We'll go into all of these in much greater depth next week. The respiratory system starts off with the anatomy and then kind of goes towards gas exchange and things like that. So I'm just going to introduce you to the upper respiratory tract, save the lower respiratory tract for next week, as well as gas exchange. The upper respiratory tract has 3 functions. It is to warm, humidify, and filter air. It is made up of the nose and nasal cavity, the paranasal sinuses, which aren't shown, as well as the pharynx. The functions of the nose are to moisten and warm air, filter and clean inspired air, serve as a resonating chamber for speech, and houses olfactory receptors. And we'll talk about kind of this a little bit more in depth. In the nasal cavity, we have hairs, and those hairs are going to filter particles from the air. These hairs are going to, this is kind of like the first filter that's of air coming in. And then there are 2 sets of mucous membranes. There is olfactory mucosa and respiratory mucosa. The majority is respiratory mucosa, but this small amount of olfactory mucosa is going to dissolve smells into the mucosa, and then the nervelets of the olfactory nerve comes in through the cribriform plate, if you remember this, through that ethmoid bone, and they hang down into that nasal cavity and sample what's going on in the olfactory mucosa. I don't think you have to know the bones that are constructing each of these cavities. If there's a particular bony structure, I will identify it. But in general, like if you read your book, it's going to talk about how it's made up of the frontal bone and the nasal bone, and the roof is made up of the ethmoid and the sphenoid. That was more 1147. You're like, yay, done with that. So you definitely need to know those functions. You need to know the purpose of having an olfactory mucosa versus a respiratory mucosa. A respiratory mucosa is going to have its pseudostratified ciliated columnar epithelium to move the mucus into the throat, which will be swallowed and digested. And it will contain goblet cells, as well as something called seromucous nasal glands. Seromucous nasal glands produce 2 fluids. They are going to form a serous fluid, which is watery, containing enzymes. The 2 main enzymes are lysozyme and defensins. Both of these are going to be antibacterial, is the main one. And then the mucus cells are, it's going to be so that it's thicker and will help in the moisturizing and the warming of the air. So we have a thick mucus secretion and then a watery secretion that contains protective enzymes such as lysozyme and defensins. Nasal mucosa also is responsible for the sneezing reflex. I think these might be my favorite nasal structure, are the nasal conchae. These are squirrel-like, they're, picture? Okay, they're these, okay. So there is a superior, a middle, and an inferior nasal conchae, and they form ridges on the septum walls. So they line the nasal cavity, and what they do is they create turbulent airflow. So we suck air in, and then it needs to spend some time in order to get warmed and moistened and filtered. Okay, so the nasal conchae slow everything down so that it can get properly, filtered and warmed and humidified before moving on. So I love the nasal conchae. They're really cool. And the interesting thing too is that when air is exhaled, they will reclaim some of that moisture and warmth in the heat too, so we don't lose it all. So nasal conchae are very important structures. During inhalation, they filter heat and moisten air, and then during exhalation, they reclaim heat and moisture because they're creating this turbulent flow. Because they slow things down and filter so well, they filter out anything that is larger than 6 microns. Just for reference, like coronavirus is 0.1, so not going to filter that out, unfortunately. The sinuses, these are air-filled spaces located within bones. If you remember, they are located in the frontal bone, the sphenoid bone, the ethmoid bone, the maxillary bones, and also the temporal bones, but that's not part of the respiratory system. So paranasal sinuses are going to lighten the skull, secrete mucus, which then drips down into the nasal cavity, and they'll also help to warm and moisten air. Pharynx is the last structure of the upper respiratory tract. It's about 5 inches. It's a muscular tube. It is made up of skeletal muscle. It runs from the base of the skull to around C6. There are 3 regions: nasopharynx, oropharynx, laryngopharynx. The difference in the nasopharynx is that only air is going to pass through. So the nasopharynx is the upper segment. So this upper segment. Then we have the oropharynx is this middle segment. And then the laryngopharynx is down here by the larynx. The nasopharynx should be only for air. And then during swallowing, the uvula moves up and blocks it off. But if people start to laugh, this becomes leaky, and that's why, like with little kids, like milk will go out their nose because this didn't seal correctly. But normally, the nasopharynx should be just for air. The oropharynx is going to be a common passageway between food and air, as is the laryngopharynx, and laryngopharynx continues on with the esophagus. Okay. The pharyngotympanic tubes, your auditory tubes, they also come in at the nasopharynx. The nasopharynx, because it's only for air, has pseudostratified ciliated columnar epithelium because it still needs to move mucus down to the oropharynx. The oropharynx, we're going to switch over to a stratified squamous epithelium because it is more protective, and it is the passageway for food and air. The laryngopharynx is also a passageway for food and air. It's continuous with the esophagus, and it is going to have stratified squamous epithelium. So this one, naso, is more like a nasal cavity with pseudostratified ciliated columnar epithelium, whereas the oro and laryngopharynx are going to have stratified squamous epithelium to be more protective as they move down towards the esophagus. And that is where we stop, and we'll pick up next week at the level of the larynx.